All right, let's dive right in. Today, we are unlocking the very code of life itself. We're talking about a field that's changing everything we thought we knew about evolution, disease, and what makes us, well, us. This is all about comparative genomics.
So let me just start with a question for you. Why on earth would a tiny fruit fly be one of our biggest secret weapons against human cancer? It sounds a little crazy, I know, but the answer reveals this absolutely incredible connection between us and this tiny insect. It's basically a genetic detective story, and all the clues are found by comparing the blueprints of life.
But, before we can even start comparing these genetic blueprints, we have to talk about how we learned to read them in the first place. And that journey, it's pretty amazing. Okay, just look at this timeline. The speed of this revolution is just mind-blowing. Back in the 90s, the Human Genome Project was this massive international effort. It took more than a decade and cost billions of dollars just to read one person's genetic code. But then, fast forward to 2010, new tech just completely shattered that reality. That decade-long project, suddenly, it's something you can do in a matter of hours.
And this number says it all. That multi-billion-dollar project, yeah, today you can get a whole human genome sequenced for less than $100. $100. This isn't just a small step. This is a quantum leap. It's the very reason a field like comparative genomics can even exist on the massive scale it does today.
So, okay, we can read the code. We can do it fast. We can do it cheap. Now what? What do we do with all this information? Well, think of it like we've discovered a genetic Rosetta Stone. So what is comparative genomics? At its heart, it's basically a high-tech game of spot the difference. Scientists use these incredibly powerful computers to line up the genetic codes of different creatures, side by side. They're hunting for the parts that have stuck around for millions of years of evolution, and also the unique parts that make each species distinct.
Right. So let's take a simple example. We line up our genetic blueprint with a fruit fly's. The goal here is to find the genes that are so fundamentally important to life that they've been conserved, or kept, across these huge evolutionary gaps. You can almost think of them as the master switches for life itself, the really critical, non-negotiable parts of the instruction manual.
But it's not just about spotting what's the same, right? It's a whole systematic investigation. It's like being a detective, following a trail of clues. So let's walk through that process.
Okay, so here's the workflow. Step 1, you sequence the genome. You get the raw data, all those A's, T's, C's, and G's. Then comes maybe the most crucial part, annotation. This is like taking an engine apart and labeling every single piece so you know what it does. Only after you've done that can you move to step 3, the big comparison across species, looking for what scientists call orthologs. These are the shared genes we inherited from a common ancestor, the real family heirlooms of the genetic world.
And this is where things get really fascinating. You'd think a bigger genome means more genes, right? Nope, not at all. Look at this chart. A simple little yeast cell has almost 500 genes packed into the same amount of DNA space where a human has just 11. 11! Our genome is way less dense. It's full of these huge stretches of non-coding DNA. It really proves that it's not just about what genes you have, but how the whole thing is organized.
All right, we've seen the how. Now for the really exciting part, the what. What have we actually found by cracking this case?
And here it is, the answer to that question from the beginning. It turns out, humans and fruit flies, we share about 60% of our genes. Now, that's not just some cool trivia fact. This discovery has absolutely massive implications for medicine.
And here's why that matters so much. Get this. About two-thirds of the human genes we know are involved in cancer have a direct counterpart in that humble little fruit fly. Now, why is that a game changer? Because a fruit fly's entire life cycle is about 10 days. This means scientists can study these genes across hundreds of generations in a short time. It lets us run experiments at lightning speed, giving us vital clues into how these complex diseases work.
But it doesn't stop with flies and cancer. This stuff gets even cooler. By comparing the genomes of almost 50 different bird species, researchers found this whole network of genes that lets birds sing. And what's really wild is that a very similar network is deeply involved in our own ability to speak. The song of a bird and human language, linked by our shared genetic history.
And the impact isn't just in medicine, it's literally on our dinner tables. By comparing cattle genomes, scientists have found the exact genes that help cows produce richer milk. And even more amazing, they found a natural mutation in a gene called myostatin that can literally double the muscle mass in some cattle.
Think about what that could mean for feeding a growing planet.
So all this incredible progress just opens the door to even bigger questions, right? We're starting to ask, what are the specific genetic changes that truly make us human? How can we go from just understanding genetic diseases to systematically curing them? And what secrets are hiding in all that so-called junk DNA that we're only just beginning to appreciate?
You know, this quote here really just nails it. The point of all this isn't just to hoard data. It's about getting a much deeper understanding of how life works, all with the ultimate goal of improving our own health and well-being.
Think about it. We've gone from struggling to read a single sentence of DNA to comparing entire libraries of life's instruction manuals. That is an incredible amount of power. And with that power comes huge possibilities, but also huge responsibilities. So the question I want to leave you with is, what do we write in the next chapter?